Good afternoon, everyone. Welcome to the ESDR Kitchen Sweet and Sour episode, where two protagonists will debate a topic in the field of skin science. This episode will take approximately half an hour and will be moderated by Sandrine Dubrac from the Medical University of Innsbruck and me, Remco van Doorn, from Leiden. We have a really interesting topic today. It concerns the beneficial and harmful effects of sunlight. This topic has received attention not only in the scientific literature, but also in newspapers and other media outlets. Even in the gloomy days of November, it is relevant to public health campaigns and how patients at risk of skin cancer are informed. Does skin light have beneficial effects in addition to promoting vitamin D synthesis? And might these effects even outweigh ultraviolet radiation's detrimental mutagenic effects on the skin? We have two outstanding speakers, both leaders in this research field. David Whiteman from Brisbane, Australia, who will emphasize UV's harmful effects. I want to remind you that in his time zone, it is now 9:30 p.m. And Richard Weller from Edinburgh, Scotland, who will illuminate the beneficial effects of sunlight. After each of these experts have given their 10-minute expose, we will have 10 minutes of discussion. And please submit your questions using the chat or the Q&A function in Zoom. It is a pleasure to introduce the first distinguished speaker, Professor David Whiteman. He is a medical epidemiologist with a special interest in the causes, control, and prevention of melanoma in skin cancer. And he received his medical degree from the University of Queensland in Australia in 1991 and his PhD in cancer epidemiology in 1997. He was awarded a fellowship in public health medicine in the same year and took postdoctoral studies at the University of Oxford before returning to Brisbane to pursue his career in cancer control. Dear David, could you please share your slides and present your case for the, I think, harmful effects of sunlight?
Yes, so thank you very much for that kind introduction. I'm hoping you can see my slides now. So as we've heard, I'm here to persuade you that UV exposure is harmful. So in our debate today, I'm up against a worthy opponent, and we're addressing this question of are there harms and benefits of UV, and what is the relative and respective balance of harms and benefits? So my opponent is going to try and convince you that there are benefits. So I want you to think about, what do we know about the evidence for causality for these associations? What do we know about mechanism? And what do we know about magnitude of those effects, of those associations? So let me start by opening the case for the harms of UV radiation. We have very strong and consistent evidence from hundreds of years of clinical observation, at least 7 decades of epidemiological data. We have very good animal models for UV-induced melanoma and squamous cell cancers. And since the sequencing of the human genome, we have very, very powerful molecular evidence that both UVA spectrum and UVB spectrum, or those wavelengths of the solar spectrum, cause photolesions in DNA that lead to cancer. So I think people on the seminar will be very familiar with the harmful effects of UVB. It's a potent genotoxin, and it causes characteristic photolesions in that DNA sequence, cyclobutane pyrimidine dimers and 6,4 photoproducts. And these are quite mutagenic. We also know that the UVA wavelengths also cause those characteristic cyclobutane pyrimidine dimers, those CPDs. They do that, ironically enough, through an interaction with the protective pigment melanin. But UVA also causes oxidative damage to the DNA helix, causing large, bulky lesions. And the problem we have as living organisms is that those photolesions in the DNA accrue. So our normal skin cells are exposed every day to ultraviolet radiation, almost every day, certainly in my part of the world. And we accrue those photolesions. We have exquisite DNA repair mechanisms, but those mechanisms are not 100% perfect. And if they're not repaired, and those cells divide, then those photo lesions become fixed mutations or lead to fixed mutations. And if this occurs again and again and again throughout the lifespan, eventually we realize the malignant phenotype in those skin cells and cancers form. And this is a very common event, as you know. We know through the work of Boris Bastian and Hunter Shane, we know which those very specific molecular pathways through which melanomas are formed. We also have very good information for the evolution of SCCs. Time prevents me from outlining all of those for you, but we know the driver mutations and we know that they're caused predominantly by UV exposure. So in that very brief opening, I hope I've convinced you that UV radiation unequivocally causes DNA damage. It occurs from both exposure to ultraviolet A radiation and ultraviolet B radiation. That damage accumulates and leads to mutations, and those mutations over time can lead to neoplasia and skin cancer. But I know and I agree that the effects of ultraviolet radiation depend on who you are and where you live. Over the course of human evolution, over the last hundreds of thousands of years, humans have evolved pigmentation in their skin to protect the skin from the harmful effects of UV. This is probably initially for protecting against depleting folate, but it now has obviously an effect for older, longer living humans. But as humans migrated out of Africa into the northern latitudes of Eurasia, they lost their pigment, and there was a very strong selective pressure for losing pigment, most likely to allow synthesis of vitamin D. Now, I just want to draw attention to where we two debaters are situated today, my opponent. And I know, Richard, I apologise. I've mentioned Glasgow here only because I have data for Glasgow and not specifically for Edinburgh, and I'm based in Brisbane. But the Indigenous populations, ancestral populations close to the equator have that deep pigment phenotype through different genetic mechanisms protecting their DNA. The Indigenous Australians have that dark pigment as well, where I am in Brisbane. The indigenous inhabitants of Northern Europe have much lighter skin. And just showing you some data here, looking at the UV index, the maximum UV index throughout the day. Here's the data for Glasgow for a 4-year period in the 1990s. So you can see that for about 5 months of the year, there's almost no measurable UV radiation at all. And then for short periods during the day, the UV index climbs above 3 or 4, and then for a short period in summer, in the middle of the day, it might reach 6. Where I'm located in Brisbane, every day of the year, the clear sky UV index at noon is greater than 3. It approaches 4 in winter, and in the middle of the summer, it's around 12, 13, 14 UV index. And so the challenge is that when we have populations that have evolved in this low UV environment, migrating to a high UV environment, such as Australia, the DNA repair systems are overwhelmed. So when Australia was first colonized by Europeans a couple of hundred years ago, they took very sensible precautions against that new tropical environment. But that all changed in the 1930s with the popularization of tanning. And so we saw a rapid increase in mortality, that is death from melanoma. This is on a logarithmic scale from the 1950s through to the 2000s, showing these incredible increases in deaths from melanoma, both in men in black and for women in white. But it's not just deaths that we have to worry about, it's actually the costs of treating and diagnosing and managing these conditions. If you take a scale of how much we spend in Australia on treating and diagnosing cancers, the UV-related skin cancers total around $2 billion a year. In fact, I've just seen the most recent figures for 2024. It's now $2.5 billion. We spend more on treating and diagnosing skin cancer than any other single cancer in Australia. It's a huge strain on our budget. But it's not just Australia that's had a problem with rising melanoma mortality. Here are the data for Scotland, showing that over the last 50 or so years, or over a space of about 5 decades, there was a more than 5-fold increase in death rates for melanoma amongst men and similar off a lower base in women. And similar data pertain to other European populations. And then looking at SCCs, for which, these are very common cancers. Rates are rising in European countries wherever the data are being kept over the last 3 or 4 decades. Rapid increases in SCCs of the skin in both men and women here in the Netherlands, and again in the Nordic countries, about a 3 or 4-fold increase in some of those countries over time. And all of these cost money. They clog up hospital waiting rooms, they clog up dermatology outpatients, and they impose a burden. So I hope I've convinced you that skin cancer is a big burden in Australia. That burden is actually being seen in other countries, both through costs and also to some extent in mortality. And the tragedy is that these are very preventable cancers. We know the cause and we know how to prevent these, and yet this problem didn't exist 5 decades ago, and it's become a much bigger problem in recent times. So you're going to hear from my opponent very shortly. So I want you to reflect again on causality, mechanism, and magnitude. I don't know exactly what Richard's going to say, but I suspect some of this data will be ecological or correlational. There may not be many measures of personal sun exposure. And I guess what the missing piece often is going to be this mechanism of action. How do we know that we can act on a cause to change the burden of disease? I will concede, of course, that we know that sunlight is very important for maintaining vitamin D, probably has a role in preventing multiple sclerosis and also in myopia. But I would contend that we really still have to consider these harms of UV. Again, it depends on who you are and where you live. In my part of the world, for people of European ancestry, trying to moderate your sun exposure is like trying to sip water from a fire hose. The UV is so intense, so strong, that it's very hard to moderate your exposure for unprotected skin. And so in our part of the world, the burdens of skin cancer are very high. I will stop in just 1 second. But I do want to bring attention to my colleague and good friend, Rachel Neal, who's done magnificent work in Australia, recognising the increasing diversity of our population. So no longer is the Australian population predominantly Anglo-Celtic. 50% of Australians were either born overseas or their parents were born overseas. We have a much more diverse population, and many of those people are at low risk of skin cancer. So we actually, and Rachel has led this work, now have new advice. I won't go through all of this chapter and verse. I would direct you to her first author publication. But essentially, for people of European ancestry, the advice remains: protect your skin when you're out in the sun. For people of intermediate and low risk of skin cancer, there are other considerations that must apply. And so I just will close out, though, and say that I would contend that even in a very northern latitude, based on current knowledge, I would say that the harms of UV through causing skin cancer, both mortality and costs, probably outweigh any benefits they might have on these other endpoints. And I say that because our public health interventions still have a long way to go and are more proximate and address the known causes of these other diseases. So we can still modify diet, modify physical activity, address smoking, alcohol, blood pressure. It's very likely that for things like multiple sclerosis, which are quite rare diseases, genetic screening will allow us to target high-risk individuals and tailor prevention. And so in closing, I would say that a policy to increase exposure to sunlight, even in European populations, will almost certainly drive up rates, mortality, and costs of skin cancer, but probably have no measurable benefit on these other endpoints. And so on that, I will close, and I will leave it for my colleague to convince me that I'm wrong, and I'm sure he will do a very good job. Thank you.
Thank you very much, David, for this very interesting presentation and point of view. So now we are going to listen to the other point of view from Richard Weller that I'm going to introduce very shortly. So Richard Weller is an academic dermatologist at the University of Edinburgh with a particular research interest in the effect of sunlight on systemic health and a clinical focus on eczema. Before becoming a dermatologist, he trained in internal medicine in England, followed by a year working as a medical registrar in Cairns, fourth North Queensland. And he then trained in dermatology as St John's in London and in Scotland, followed by 3 years full-time research in Edinburgh, Düsseldorf, and Pittsburgh. So, Richard, the stage is yours, and we are looking forward to hear your counterpart.
Right, well, look, thanks. And David, thank you. Look, I agree with 80%, maybe 85% of what David says, but I want to put the opposing or the counter view here. So the first thing to say is, look, sunlight harmful or beneficial. So here we have a picture on the left of Australia midwinter, and on the right, a picture of Scotland midsummer. The observant amongst you may notice some differences. And 30 years ago, this handsome young fellow on the left, that's me, was a medical registrar in Cairns at the height of the very relevant for Australia slit, slap, slop campaign, you know, avoiding sunlight because of the real concern, absolutely correctly, for skin cancer in Australia. And Cairns, where I was working up here, so, you know, the UV index midwinter is 7. And in midsummer, it's 14 for about an hour in the middle of the day. So we measured the UV index in Scotland in Glasgow. And this is 2 years ago, the peak UV index in Glasgow. So the UV index in Glasgow hit 7 for about 20 minutes in 2023 on the 24th of June. That was a very sunny year. Last year, it was 5 minutes. So we have kind of, you know, 5 to 20 minutes in a year of your midwinter level in Australia. So these are very different UV environments, and they have very different approaches needed to sunlight. But I want to talk about what happens here in Northern Europe in terms of sunlight and overall health, all-cause mortality. So one of the first people to look at this was Pelle Lindqvist, who is an obstetrician in Sweden. And he went in and he looked at the melanoma in southern Sweden study. So this is a cohort study looking at risk factors for melanoma in Sweden. So in 1990, 30,000 Swedish women were recruited. And they were asked about sun exposure habits, you know, sunbathing in summer, sunbathing in winter, foreign holidays, use of tanning beds. Now, obviously, lots of confounders associated with this. If you go on foreign holidays, you're richer, so you're probably better educated, so you're less likely to smoke, etc., etc. But this was extensively adjusted for. You know, lifestyle factors like exercise and smoking were looked at, social factors, income, education, health factors, pregnancy. So really well adjusted for these confounders. And they went back 25 years later, or Pelle went back 25 years later. And what he found in these Swedish women was dose dependently, the more sunlight they had, 0 to 4, the less likely they were to be dead from any cause. In fact, those that got the most sunlight, David covering in eyes in shock here, were half as likely to be dead as those that had the least sunlight. And it was particularly caused by a reduction in cardiovascular death. And remember, you know, exercise is accounted for here, and smoking. Pelle went back and did some further analyses, very interesting. He later looked, so this was a white-skinned population. He looked at the redheads, and he found that those Swedes who get the least sunlight, those that score 0 or 1, the redheads have a survival advantage over the non-redheads. But when he looks at Swedes in general, who get, you know, high and low sunlight, those redheads lose that survival advantage. And this shows how pale skin, the palest of the pale, it's an adaptation to lack of sunlight and gives a survival advantage, but only in a low light area. The other thing he looked at was the size of the effect, and he found that non-smokers who avoid sunlight have the same mortality as smokers who get the highest sunlight level. So this gives you an idea about the size of the effect. The reductions in mortality associated with increased sunlight exposure are as big as the reductions in mortality in non-smokers. Well, that's Sweden, bijou little country. So, I thought, right, let's get in there and let's get some decent data. So the UK, much bigger study. So what we've got here is the UK Biobank, 500,000 people recruited, finishing about 15 years ago, 40 to 60-year-olds. I was one of them, recruited all over Britain, and a mass of data taken. The assessment when people were recruited took about 3 hours. Lots of measures taken, bloods, all sorts of things. And questionnaires. And within the questionnaires, we wanted two independent measures of sunlight exposure. So the measures we used were sunbed users, not so much because they use sunbeds, but sunbed use is a behavioral marker for sunlight exposure. We know that sunbed users sunbathe more, seek the sun, And the other one was how far south people lived. Now, of course, you have to adjust extensively for confounders, which we did: age, BMI, smoking, social factors, deprivation, exercise. The important thing here is that the confounders move in different ways for our two different measures. And that means you can triangulate the confounders, and it makes it unlikely that an unmeasured confounder accounts for your results. What did we find? Well, the first thing is vitamin D is a biomarker for sunlight exposure. It prevents rickets, doesn't do much else, but it's a great sunlight biomarker. And we found that our sun seekers, our sunbed users, had higher vitamin D levels. So they do get more sunlight. Mortality, that after 12 years, they had a 15% lower all-cause mortality. That was particularly cardiovascular, but also cancers and other causes, multiple sclerosis, diabetes. Their life expectancy after 12 years was 50 days longer. They had an increased incidence of melanoma, as we know. Sunbed use is a risk factor for melanoma incidence. No change in mortality, but numbers are low. Other cancer mortality reduced. That was our first marker. Our second marker was how far south people live. So look, Glasgow, Nottingham, Truro, the further south you live, the greater UV exposure, and dose dependently, the further south, higher vitamin D levels. So we know this is a good measure for sunlight exposure. So the difference between living in Glasgow and Truro in Cornwall, 12% reduction in all-cause mortality, particularly cardiovascular, also cancer. Yes, 16 days longer life expectancy after 12 years for every 300 kilometers south. Increased incidence of melanoma, no change in mortality, other cancers down. So we published that last year. Work we're doing at the moment and about to submit, we've gone in and got much more detail, and we've incorporated the other sunlight measures in the biobank. So we've looked at, solarium use and where people live, how much time they spend outside, more or less than 4 hours, and also sunscreen use, because in Britain we find that sunscreen use observed is a marker for sunlight exposure. So people get a score of 0 to 4 for how much sunlight they've had. Every point increase gives you an increase in vitamin D, so we know this is a sensitive indicator of sunlight exposure. And we can classify people in the biobank into low, medium, high sunlight exposure. We can then look, after adjusting for confounders, at their mortality, and dose dependently. So green, no change, is low sunlight. Blue is medium sunlight, red is high. Dose dependently, the more sunlight people get, the lower their all-cause mortality, particularly cardiovascular, also cancers other than MS. An increase in them, but not dose dependently in melanoma. And look, the oldest person now is 75. SCC deaths in the UK don't really start until people are over 75, almost unknown below that age. Peak mortality from SCC is in people's 90s, so we don't see that pattern yet. We can now calculate a population attributable risk for sunlight and mortality. Yes, 9% of melanoma deaths in the UK can be attributed to sunlight. However, it's the other way round for all-cause cardiovascular and cancer mortality. We have a reduction in death from these diseases with increased sunlight, and we can now calculate the risk-benefit ratio. So there are now 35,000 deaths in the biobank. We're now 15 years after it closed. When we multiply the deaths from these different causes by the population attributable risk due to sunlight, we found of the 400,000 people we included in the study, there are about 40 deaths from skin cancer caused by that could be attributed to sunlight, 1 in 10,000. However, a much greater number of deaths which can be attributed to insufficient sunlight. If in the UK we reduced population sunlight level enough to prevent 1 death from melanoma, it would cause 75 excess deaths from other causes. So I'm just going to finish off just the Bradford Hill criteria. Bradford Hill first identified the link between smoking and lung cancer. When you're trying to attribute causation to observational studies, it's complex, as David points out. These are the 9 factors he identified as being important. Strength of association, Pelley's smoking effect size, our 75-to-1 ratio, really strong. Consistency, there's up here in Northern Europe, there's us, there's Pelley, there's also the Swedish lifestyle and health cohort. There are 3 cohort studies in Northern Europe. All of them show reduced mortality with increased sunlight. That may not be the case in Australia. It may not be the case in America, but consistently it is here in Northern Europe. Specificity, I haven't got time to show this. Some conditions are reduced. Some, there's no effect of sunlight. It has specific benefits. It's not snake oil that does everything. Temporality, obviously these are cohort studies. Sunlight exposure comes before the outcome. We have a biological gradient, a strong dose-response relationship. Vitamin D doesn't do very much. We have nitric oxide pathways, we have immune pathways. We're working on that now. Nothing's been done for 90 years. No one's looked at it. It's always been the bad. We know about seasonal and latitude variations, coherent with that. There's no experimental data. There's no clinical trial data on smoking and lung cancer either. And it's analogous with the vitamin D data we know as a marker for sunlight. So sunlight, harmful versus beneficial. Well, it depends where you are. You know, Australia or Scotland, these are very different. It also depends on whether you're looking at systemic health, benefits really strong, or skin health. It has adverse effects, and skin color really matters. So there we are.
Thank you so much for sharing your insights both. I would like to invite the audience to post questions and comments in the chat function, but I think for now it would be most interesting to use the next 10 minutes for you to perhaps discuss or respond to each other's provocative statements. I think that David Whiteman's position is the least provocative of the two. And Professor Weller, maybe may I invite you to comment on the statement that a large proportion of melanoma and other types of skin cancers is attributable to UV. Do you share this? Do you agree with this view, or is the proportion of 50% or more attributable an overestimation? I hope I'm summarizing part of your presentation correctly.
Yeah, so look, so squamous cell skin cancer is caused by sunlight. We have dose dependency, chronically sun-exposed sites, absolutely right. In the UK, we don't really see SCC deaths until over the age of 75. In their 90s is the peak. Average lifespan in Britain is 83 for women, 81 for men. Peak death from SCC is in their 90s. My concern is that dermatologists, in their desperation to prevent deaths from SCCs, would rather exchange death from SCC age 90 with death from a myocardial infarction age 60. That may be a dermatological success, it's not a medical success, and that really concerns me. The melanoma attribution, 90% of melanomas in Britain are caused by sunlight. This is based by calculating incidence now with incidence in a historical cohort. Australia is the only place that's done good data, actually. So the problem we have is there is undoubtedly an epidemic of overdiagnosis of melanoma. Huge rise in cases, much greater than the change in mortality.
Thank you.
So
Professor Whiteman, in addition to, would you like to add something to this regarding your personal presentation, and more specifically, could you perhaps comment on the claim that the mortality is can be the risk of all-cause mortality can be reduced by sufficient exposure to sunlight?
Sure. Well, just on the first question about melanoma specifically, I agree that melanoma has a complex causal pathway. I think, though, that it's very likely that almost all cutaneous melanomas have their original causal mutation in the melanocyte in early childhood, probably from sun exposure, but probably not from an excess of sun exposure, just bad luck. UV photons hitting a dividing melanocyte, but we have very good data that it's actually early life exposure, both in Australia, US, Europe. Early life sun exposure is likely the initiating causal event for melanoma. And then whether or not you get a melanoma, when it expresses in middle age, many of those people have a genetic background that predisposes them to melanoma, as in a nevus-prone phenotype and fair skin, and they just happen to have had a bit of sun exposure as a child. So we know that protecting children lowers their nevus counts and lowers their risk of future melanoma. Just in terms of the mortality data, I agree they're very intriguing data. I have seen the UK Biobank paper. I would say that there's still an open question about how much residual confounding there is. I appreciate a lot of adjustment has happened, and of course we have to with these sorts of observational data. I think solarium use as a marker of UV exposure is a very particular kind of exposure. It's very particular kinds of people that use solaria. They tend to be a different cluster of personalities, predominantly, I think, in the UK and in the biobank as well, predominantly younger females are the main users, or were the main users. And so all of these factors do confound these associations. I can't recall having seen sex stratified analyses. Maybe they've been done.
We did all that. Yeah.
So I think these are intriguing findings. They're definitely worthy of pursuit. Whether or not they're causal, I think it's early days, and we know that there are other very strong risk factors for those various high fatality conditions that are contributing to the UK mortality burden. And I guess just sort of anecdotally, mortality from cardiovascular disease and other major killers are fairly similar between the UK and Australia, apart from these things like melanoma and skin cancers. So the difference in population exposure wouldn't seem to be driving much lower rates of cardiovascular disease or other cancers in Australia. So, but that's purely anecdotal. It's not epidemiological. So I don't want to rest my argument on that rather weak proposition.
There are 4 questions in the chat, or 3, perhaps to both of you. So the first is, were the Swedish studies, I think that is for Professor Weller, adequately corrected for non-exercise activity levels?
Well, they were. I mean, there were. It was a questionnaire-based study, and it was corrected for exercise with all the data. So yes, it was adjusted for exercise. I might say the biobank study incredibly extensively adapted for exercise. So not only do we have what people reported as exercise in the biobank, they then went back to the biobank and 50,000 people wore movement monitors to then calibrate the reported exercise with actually observed with movement monitors. That was then fed back into the biobank, because when you publish data in the biobank, you put it back in for other people to build on. So we have incredibly detailed data on exercise in our biobank study. And exercise is unbelievably good for you. You know, exercise and sunlight and not smoking, I would say, are the 3 most important things you can do for your health. So yeah, but you know, Pele also adjusted for exercise.
The second question that's also for you is, do you think the benefits of sun exposure are exclusively related to high levels of vitamin D?
No, hardly any of them are caused by vitamin D. Vitamin D's benefit. So we know that high vitamin D levels associate with better health of every kind, not of many kinds. However, we now have huge numbers of clinical trials of vitamin D supplementation. So the VITAL study in America 25,000 people, 5 years. The D Health study, led from Queensland, I think.
Rachel Deal. Yep.
Rachel's amazing study, 21,000 in Australia, really important study. And these were for 5 years, and they ended up showing that vitamin D supplementation does pretty much nothing. A very few minor effects, autoimmune disease, not very much. The difference in those trials showing vitamin D is a bit of a damp squib, and these incredibly strong associations. And the problem is that vitamin D stuff has blinded us for the last 80 years. Oh, sunlight's benefits, it's vitamin D, take vitamin D. It's not the vitamin D.
Do you think it's nitric oxide or maybe anti-inflammatory effects of immunosuppressants?
Yeah. So the nitric oxide pathway, really important in cardiovascular health. Look, I'm cited because I showed that, but so did Christoph Sustrek in Düsseldorf, independently has validated, and so we've both independently shown sunlight releases NO has cardiovascular benefits. It's also been done by other groups in Britain. So undoubtedly the NO pathway really important. I haven't had a chance to talk about it today. We're going to be submitting to the ESDR for next year's meeting. We have some fantastic, really exciting work that we're about to submit. We have found, together with Amaya Virus in Manchester, an UV-driven, really important immune pathway which reduces that cancer mortality, we think. I mean, hugely important. You'll be hearing more next year. So it's another mechanism for David.
If I may, what type of study would now be needed to elevate this correlative data with UV, linking it to cardiovascular disease in particular, to a more causal and convincing, quality?
So I might say we're doing the studies. I've only had 10 minutes today. We've done studies where we irradiate people for 2 weeks in winter. So we give people UV in winter, we then take measurements of them. Amaya Virus in Manchester is irradiating mice and doing kind of cancer models in that. We can also then cross-reference our irradiated human data, outcome measures like proteomics, with observational data in the biobank. So we have the same measures with natural UV exposure and artificial UV exposure. And we can cross-reference our single-cell sequencing and spatial transcriptomics data in our irradiated humans and our irradiated mice. We've got a lot more coming out driving these mechanisms, because until they're there, I think it's hard to persuade people.
David,
and I think just adding, yes, I would like to, and I think I applaud Richard for undertaking all of those experiments, because I think they're exactly the kinds of data that people would want to see. But again, these address intermediate endpoints, things like blood pressure or heart rate or other things. And I think what we would like to see, and it would be very hard to do, but to have studies where we have personal UV dosimetry on maybe tens of thousands of people, and then follow up over time looking at cardiovascular events and other events and mortality from those events. They're costly, hard studies to do. So it's a level of evidence that will be quite demanding to reach, but that would be persuasive as well, I think, in addition to these intermediate endpoints.
And I might throw in, you can't just throw a UV monitor. You need to really well design it, because if it turns out that runners get lots of sunlight because they're out running, and computer gamers get no sunlight, well, you've got to design the experiment so it's not confounded by all of this.
I completely agree, and observational data will always have flaws, confounding, and it goes both ways. So, and this can bedevil any of these sort of causal discussions, because observational studies are mired in confounding, particularly for these long-term follow-ups with complex endpoints and for conditions that have complex etiology. We have to consider all the other causes.
Yeah, thank you very much for these very interesting discussions. But now we have to come up to the end of this episode. I'm sure that the discussion will going on later. And then it's very stimulating discussion, actually very, very, very interesting. And the role of vitamin D for sure is not yet fully uncovered. The next, we would like to bring your attention to the next ESDR Kitchen, that will be an EISID flavors about environmental influences on dermatologic diseases, given from Yumi Matsuoka, Nakamura, and Jan Miles. I would like to emphasize that this ESDR kitchen will take place at 3:30 p.m. CET instead of 12:30. So please really take note of this date. And this to remind you as well that all the ESDR episodes are available online via YouTube, either via the ESDR website. So this being said, I would like to thank you very much for this very nice presentation, and maybe give the last word to Remco. Thank you.
Well, I have very little to add. Thank you, Sandrine. I would like again to thank the protagonists here, so Professor Weller and Professor Whiteman. I was surprised to see that you have a lot of shared opinions, more than I'd expected, and I'm really looking forward to seeing you resolve the issues that we discussed in the coming years with the studies that are ongoing. So thank you as well, Sandrine Dubrac, and thank you to the audience. I'm sorry that we have not been able to deal with all questions that have been posted in the chat, but perhaps this can be attended to through email. I will see if this is possible. So thank you again, and looking forward to the next ESDR Kitchen webinar. Goodbye. Thank you.